or the second half of this morning's session. So I'm a biophysical chemist, and I come from a protein structure, protein biophysics background. So my presentation this morning will be a little bit different flavor than what we've heard so far. So, and but what I'd like to talk to you about is a problem in trying to understand trying to provide tools for biosimilarity between complex protein therapeutic drugs. So I'm from the National Institute of Standards, and this came to our attention both from our we're an agency that supports commerce and industry, commercialization, as well as other U.S. government agencies to provide harmonization and standards and validation of methods. And so this problem came to us first from the FDA, but from other regulatory agencies, that there's a large sort of wave of biologics that you all may be aware of that are coming down the pipeline for approval in the U.S. and EU and around the world. And and with the expiry of many of the patents, or the so-called patent cliff, there's a number of biosimilars and biobetters being developed. And so there's this sort of question about what analytics can be used to move these new complex drugs and sort of copy drugs into the market through a regulatory approval. So, yep.
Sorry for my ignorance. Can you please tell what's the difference between biobetters and biosimilars?
Okay, so the goal often in this is analogous to making generic drugs. So the goal is to make a drug which is the same as the one that's already been approved, FDA approved. In the course of making some of these biologic drugs, what they found is through improvements in the biotechnology process, they've actually gotten drugs which actually have better performance. So it creates an odd problem because in science you want to do better, but to match for matching similarity, you want to match the original drug. So the question is, what do you do with drugs that are actually better performing that you want to say, or that don't have to go through a whole new series of FDA approvals?
Presently, they have to go through
Well, I'll get to that. So what the FDA at least is trying to do is to create a pathway, and this only came about with the Affordable Care Act. It was written in a regulatory pathway in the U.S. The Europeans had a pathway before us, and the idea is that to shorten the timeline, to go through the process, and to reduce the cost. Of course, what you would like to do is to have as good an analytical package on your drug as you can. And so that would actually reduce the need for clinical trials. So anything you can do to establish similarity in your product at an analytical level before you get to the clinic should in principle reduce the time and costs. So one of the things that why we've gotten involved in this, in particular for the complex biologic drugs, is that higher order structure or foldedness of the proteins are actually a critical feature, a quality attribute of those drugs, and distinguishes them from small molecule drugs where you simply can understand a drug based on its constitution and its concentration. So again, coming back to what sort of motivated this, there was a congressional hearings, and the FDA and NIST came together to talk about the need for understanding, the ability to predict potency of biological drugs would be better, of course, if you had ways to measure and quantify the three-dimensional structure. This was Steve Kozlowski, who's head of the chief at the FDA, who's been actually working and consulting with us on what would be useful to do. So the question again from a NIST measurement, we're a measurement science lab, is what are the best measurements to tell you what the structure is, and what's the confidence of those measures so that you could feel confident that when you get a measurement, you're sure that it's actually telling you what you think it's. Do you have good controls, good precision, good accuracy? So the method we promote for looking at higher order structure is NMR spectroscopy, and for those who are not familiar with it, if you look at a protein structure, as you see here on the left, this is sort of a stick diagram of a protein. It's very complicated with thousands of atoms. But if you look at the backbone of a polypeptide, which is what the proteins are comprised of, it's a polypeptide backbone, there's correlations along that backbone, particular amide correlations between nitrogen and proton, which can be measured by NMR to give a so-called spectral fingerprint of that protein. So each one of these NH bonds or atom pairs can be correlated, and each one represents a local structural chemical environment of the protein, which is unique in the folded state. So the pattern you get for the spectral positions of those correlations give you a unique fingerprint for that folded structure. So one of the things we had to do was actually apply this kind of a method to real-world applications. So with NMR, you need isotope active atoms. We had to use basically drugs that are coming off of production, so you can't use isotope enrichments. And so we had to develop methodologies that were able to be applicable at low natural abundances. And we did this through use of advanced technology in cryoprobe and high-end NMR. So as I mentioned in the previous slide, what the NMR spectra can give you is if you look at a protein structure, and this is G-CSF on the left here, it's a chemokine drug. It's actually the first accepted FDA biosimilar application that was accepted about 2 months ago as the first one in the U.S. to be accepted to go through review. So in the two-dimensional NMR spectra of G-CSF, if you look at all of these spots there, each one of those spots again represents a single nitrogen-proton correlation in a position, unique position in the protein. So each position shows in principle for one, if there's one structural state of the protein, one positional frequency dot correlating the protons on one axis and the nitrogens on the other. And this composite pattern is a fingerprint of that structure. So any alterations in that pattern in principle can tell you about alterations in structure very rapidly. So in order to validate, so one of the things we do to validate methods is we try to reproduce that method across multiple sites, multiple measurement platforms, to demonstrate that you can reproduce the same data to give confidence that when that data is then presented, it's not lab dependent or instrument dependent. So we did this by collaborating with the FDA, Medical Products Agency in Sweden, Health Canada, and NIST. So 3 regulatory agencies that we partnered with, and then NIST. Oops. So the goal again was to establish through a so-called system suitability sample. We basically would compare the results on one sample distributed to all the labs. And by doing that, we'd basically enable harmonization of the method and determine the degree to which the measurements would be comparable. Again, setting a benchline for the precision of the measurement. So here's an example of some of the data we've been able to collect on the suitability G-CSF sample. And what you can see here again is if you look at this two-dimensional plot, you can easily visually tell if two things are the same or different by your eye. And although it's very hard to see, you'd have to sort of trust me to a bit here that if you look at the different data sets they're in different colors here, it's almost impossible to see the differences because they're right on top of each other. So the NMR method again shows a high level of precision in mapping out each one of these spots. And it also shows on the spectra that with other methods, we can actually assign each one of these correlations to specific amino acids in the protein. So not only do you get a unique fingerprint, but you can map it onto the sequence in a sequence-specific way. So you can see that visually, but what we can also do is by actually doing sort of simple RMSD comparisons, we can establish the measurement drift or the measurement variation between the spectrometers and the laboratories, and that's what's shown here. Basically, we've shown that there's that the differences that we can detect between the spectrometers on the same sample on 2 different protein sort of protein examples here, G-CSF and another SH3 domain, are in the parts per billion, 3 parts per billion in proton and 11 parts per billion in nitrogen. So that may not mean much to this audience, but it basically says that the lab-to-lab, instrument-to-instrument variation is well below a precision that would require to detect any differences in these samples that would be significant structurally. So it's a highly precise method. So again, we can not only look at the precision of the whole measurement, but we can take each peak and look at its position and compare position by position along the sequence. And that's what's shown here. So if you actually look along the sequence of the protein and compare position by position sequence for each one of those correlations, you can get a correlation point to point across the whole polypeptide instead of this composite precision. And what this correlation shows that basically, again, for the first pass, most of the data was well below our instrument precisional level, but there was actually a variation in one of the sites. And this is part of the reason why you do these kind of studies, is to establish the criteria for getting these kinds of matches. And it turned out that this set of data, which came from Canada, was on a spectrometer that was miscalibrated by temperature. And so there's quite a bit of outliers because with the different temperatures, there's actually different chemical shifts that come from alterations in structure. But once we harmonized the calibration of all the instruments and the Health Canadian instruments came into the right calibration, the precision of the data, again, across all labs was extremely high. So the key to this methodology is and compared to other spectroscopies, is all we need to be concerned about is spectral resolution. Can we define each one of those peaks and map it onto our backbone so that we have a reporter at every position in the protein? And can we do this? And again, there's protocols that we are publishing to say how you could do this in a standardized way, how you handle the data, how you pick the data, how you compare the data. So this basically determines how many peaks you can see and how well the peak positions can be determined, because what we're doing is a frequency to frequency match. And the second thing is a signal to noise. So you have to sort of establish a platform signal to noise, and we have ways of correlating signal to noise to precisional correlations also that we've come up with that I won't go into today. And again, it tells you how many signals you can see. So once you establish a signal to noise requirement and a resolution, then you should be able to reproduce these spectra across any lab and any platform. And because they're frequency matched, eventually we can deposit these things in databases, and you can compare them to a reference data set without actually having to take a sample set again. So in the second round of this round robin study, we did a sort of a comparability, a biosimilarity comparability. So we looked at this NMR as a tool for testing comparability of therapeutics, which is used for lot-to-lot comparisons by manufacturers to assess manufacturing change, and then for biosimilar assessment. The G-CSF Filgrastim samples were again selected because there's an innovator product, Neupogen from Amgen, and a number of follow-on products which we're able to get from India. These are not US approved, but we could get our hands on them. So again, I said we have to be able to do these experiments on samples right out of the, or drugs right out of the vial. And so this just shows you that even when there's a very small amount of protein relative to the excipients and other molecules that are inside of the formulated product, you can see in this trace here, you can barely see the protein unless we blow it up compared to the sorbitol. There's no problem in actually collecting these 2D maps in the presence of the normal formulated drug. And again, what we can do is overlay the different drugs on top of each other and get an assessment of how close they are based on how close these fingerprints can match. And again, for these biosimilar drugs, they seem to be highly similar structurally. So one of the other things one can do with the data is to use a sort of total analysis. So what I talked about was a point-to-point comparison. If you use a principal component analysis of the data, you basically can take the entire spectra and break it down into 1D traces, which then are fed into a matrix for 1D by 1D comparisons. It doesn't have any bias that might be associated with peak picking or deciding what's a real peak, what's not a real peak. And it doesn't require assignments, and it can handle regions where there might be overlap of spectra. So it basically encompasses all the data. And so if we do that, we can again look at our data sets, and we can show the degree to which all the data sets cluster. Each data set clusters best among its particular instrument, but then you can see the spread that one might expect between the different instruments. It turns out that we again could pick up things about operator, what we call operator variation. So for the Swedish data, there was a different experimental protocol used, and so their data spread is a little bit higher than all the other sites. And we could also pick up things like the suitability sample, which was produced in-house and wasn't a drug product, actually has is slightly offset from the drug products because of the different way in which it was produced. So just in a few minutes, I just point out that when I talk about this, we've started looking at comparability of this chemokine class molecule. NMR is good at looking at relatively small proteins of 30 to 50 kilodaltons or less, but it's really the monoclonal antibody drugs that are really what's looming large on the horizon in terms of the wave of biologics and biosimilars. And so the question is there anything we can do about that using these NMR fingerprinting methods? Well, if you look at the spectra of an antibody, this is the methyl spectra and this is the amide spectra. Again, without thinking about any of the details and not knowing much about NMR, I hope you can see that these quality of these spectra are much more overlapped and broad because these are much larger, flexible molecules. It's 150 kilodaltons, and so you're going from 100 or so amino acids up to where you have a 50 kilodalton to 10 times the molecular weight and complexity. So the one thing we can do is actually assuming that the structure is embedded in these domains. There's a well understood protocol for a single cleavage digestion of monoclonals into their FC domain and FAB domains. And if we do that, we can actually regenerate these nice correlated maps, and using these kind of well-resolved correlated maps, we'll be are able now to sort of map these structural changes in those molecules. And just as an example of that, we can look at the FC region of our NIST antibody against the sigma antibody, and basically because the FC region is the constant region, there's a high degree, if we do a correlation between the two spectra, there's a high degree of correlation as one might expect. But if you look at the FAB region, which is the variable region of the antibody, specific to the antibody, then the correlation goes way down, as again one might expect if you compare the two spectra. So it's actually it's a stark contrast because we're looking at two different antibodies, but we can easily pick up the differences and the similarities. So in summary, I just wanted to convey to you today that if you think about spectroscopies that are used for analyzing drugs, NMR has a long history actually being used as an analytical tool both for small molecules and for large. It's unique in its ability to assign the resonances to the atoms in the molecule, so you get a high degree of precision and resolution and assignment of the probes to the structure. It's a complex method, but it's very easy actually to run. Once you set up the protocols, it doesn't require an expert to run these experiments. There's a very good lab-to-lab reproducibility, which is often an issue in analytical tools. The NMR spectra seem to have no significant drift between labs and across the 1 year of the study. So we can do things at natural abundance, and again, as I pointed out, we can pick up things like pH or temperature. I pointed out the temperature, so we want to make sure our conditions are right, that you don't measure things that aren't really significant. And we can do it on even large, contribute to characterizing these large MAB molecules. So with that, I would just wrap up by acknowledging the people that worked on this with me. At our institute right here in Maryland, it's joint institute with the University of Maryland, Rob Brinson and Luke Arbogast. Luke is a postdoc, Rob's a staff scientist. And then these are our FDA collaborators, David Curie, Human Gasseri, Health Canada, Eve Alban and Derek Hodgins, and then in Sweden, Ian McEwen. And we acknowledge the work with the Wagner group at Harvard for some of our NMR method development. So with that, I would take questions. If there are any questions. Yes?
Can you generate histogram from NMR signals? So on the x-axis, you would say bin the signals into groups and count how many signals belong to each bin.
Well, yeah, that's sort of a chemometric approach, right? So we did something similar to that with the principal component analysis. So what you do is we arbitrarily bin, we take our two-dimensional spectra down to one-dimensional spectra and build them into a complex matrix, and then we bin them and compare bins against each other to get the PCA analysis done.
Have you ever generated any histogram from that? Or can you?
In terms of,
If you have a bin,
yeah.
then you can count how many signals belong to each bin. Is it possible to?
Oh, sure. I mean, you could, in principle, the goal is to get the spectra resolved to the point in more than one dimension that there's only one signal per bin. That's what our we want to actually have signal to signal comparison and not to have multiple signals on top of each other. So we can have unique point to point.
You cannot generate histogram.
We could, but I don't know if it would be useful.
You
We could.
can generate histogram, and if it's a fat tail, the Planckian distribution equation may capture that and reduce that complexity into 3 numbers, which may differ from one protein to another.
We could. I mean, we don't think about it that way, but we could think, sure. All right, so any other? Yeah.